to all of you. And I just wanted to quickly check if my slide is visible. It is a blank slide.
Yes, sir, it's visible, sir.
Wonderful. Okay, so thank you, Nidya. And uh, first of all, thank you very much to all of you who have joined into this call. I see 60 raising up, and it's really a encouraging situation where a lot of you have joined into this call in the evening of a Thursday. So this seminar series is called BioEdge, and this BioEdge seminar series, this is the second technical talk. So we started off with the technical talk, and then we had a professional skills talk, and then again we are meeting for a second technical talk. Yeah, to quickly give a recap, I will show you in the next slide what are the things we covered in the uh, first talk. Before that, I just wanted to share something with you. If you are a student who are into graduation or post-graduation, studying in a college or university in any of the subjects, it could be life sciences, it could be bioengineering, whatever you are studying. Two things I would like to consider when it comes to how do you want to approach your life at academic institution and post the academic institution, how your approach has to be when it comes to career. When it comes to academia, it would be a really good idea if you can start looking at the entire thing from one more aspect, that is ROI. We call it as return of investment, which means you have invested certain amount of money, and you have investing certain amount of time, and it is a good idea for you to make sure that you are utilizing the amount of money and the time you have invested into your studies. So, you know what I mean. So you may have to look into varied aspects of your academic life. When I say varied aspects, it is not only about your core subjects, but also the other micro credentials you may have to do along with your course. Once you complete your degree or post graduation in any of the college or the university, uh, when you come out and start searching for the job, this is exactly the purpose of this presentation. That's why I wanted to talk about IOR before anything else. IOR is industry organization role, which means when it comes to the career, when it comes to joining a job, the first and foremost thing I would like you to look into is in which industry you are trying to join. Is this industry flourishing? Is this industry prospective? Is this industry posting a lot of opportunities? In the last presentation, this is one of the things we spent a lot of time in understanding what are all the avenues in biotech industry, what are all the opportunities in biotech industries, right? So we have seen different segments and different organizations and so on. So that is the industry part of it. In this industry, what is the organization you would like to work? This is very, very important. Are you uh, are you desiring to work in an organized, very large organization, multinational company, or it's a Indian-based multinational, or it's a large organization, or would you like to work in a MSME, or would you like to work in a startup? There is nothing right or wrong in any of this, right? It is a career choice. Some of you may want to join a large organization right in the beginning. Some of you may want to start in a startup, learn the trick of the trade, and then move into the medium scale or a large scale organization. So that is the organization part of it. So when you start looking into your career, the first thing I would like to look into, I want you to look into, is the industry, in which domain. Yeah, in the last presentation, we went through a lot of individual segments. So in which segment you would like to join, number one. Number two, in that segment, which organization you would like to join. The third one is in that organization, in which role you would like to join. Again, going back to the last presentation, in the last presentation, we spoke about how do you align your personality trait into your job roles. We discussed about 6 job roles, and these six job roles is what we are going to explain in detail in this presentation. All right. So basically, your alignment to your career should be in the form of in which industry I would like to work, in which organization I would like to work, and in that organization, in what role I would like to work. With this background, I again welcome you all to this discussion. As I told you, we will go through a quick recap of what we have discussed in the first tech talk. In the first tech talk, we started off with what biotechnology is. Is biotechnology is a science? Is it a faculty? As we discussed, it is a completely a multidisciplinary faculty, and it is also a interdisciplinary faculty. So if you have to be successful in biotechnology, really you should have some kind of multidisciplinary aspect in you, understanding and working with multiple teams with your core domain knowledge in you. So that is what biotechnology is all about. And then we discussed about biotech market segments and trends. What are all the different market segments in biotechnology? For example, it could be biopharmaceuticals, bioagri, it could be bioservices, or it could be bioalternative fuels, or information technology, bioinformation technology. So these are the different segments we discussed in the last uh, tech talk in detail. And what are the trends? How India is clearly aligning with the global market trends in biotechnology. And then we entered into the individual segments. In each of the segments, I told you what are the sub-segments which are available that we discussed in detail. And after the discussion, we spoke about skill composition. This is where we discussed about what is your personality trait, how to identify how you can align your personality trait to the job roles. What are the different job roles available in the industry and how to align. And finally, we touched upon it is not an individual skill, it is a skill composition. When I say skill composition, it is a composition of technical skills, professional skills, cognitive skills, physical skills, emotional skills, and social skills. So it's all put together. All together, you form a very strong skill set. These are the things we discussed so far in the first tech talk. In this talk, I would like to focus completely on the domain. When I say domain, I'm going to talk about pharma, biopharma. Though I say pharma, biopharma, majorly I'm going to focus on the biopharma. How does the pharma biopharma differ from each other, and what are the different types of medicines that are produced in the pharmaceutical industry and biopharmaceutical industry? Majorly, there are two categories. Majorly, there are two categories of drugs that are manufactured in a pharma and biopharma companies. One, it is called as a small molecule. Other one is called as a large molecule. When I say small molecule, we are talking about chemically synthesized compounds, and these are all very small in molecular weight. I'm taking example of aspirin, which is 179.15 daltons. It's not even a kilodalton. It's one tenth of a kilodalton, not even 1 kilodalton it is, right? So it's a very small molecule. It is less complex in its structure, and when it is compared to large molecule. Large molecules are very complex structures. As you can see, a simplest insulin molecule is this complex. It has so many amino acids, so many beta-plated sheets. Those who are from biotech background who are attending this talk, you will understand what I'm talking about. In the tertiary structure of a protein, you have the beta-plated sheets and coils and so on, right? So it is a mix of everything. It's a very complex one, number one. Number two, most of the times it is biologically synthesized. And the molecular weight is really very high. As you can see here, it is 5,000 Daltons. So it's a 5 kilodalton molecule. So it's a large one, large molecule. And that is how you can keep in your mind that there are two categories of drugs produced, small molecules and large molecules. Most of the times, the small molecules are chemically synthesized and it is coming under pharmaceutical industry. And large molecules are biologically synthesized. Some of the small molecules are also biologically synthesized, but if it is biologically synthesized, then it comes under the biopharmaceutical companies, biopharmaceutical organizations. For example, it could be insulin, erythropoietin, interferon alpha, and you have series of monoclonal antibodies, biosimilars of monoclonal antibodies, that is biologic biosimilars, and of course vaccines and therapeutic proteins and so on. All these comes under the biopharmaceutical organizations. Organizations produce these type of products. So that is the basic understanding about two major categories of drugs that are manufactured in the industry: small molecules and large molecule drug products. Now we will clarify one more point here, because when you learn about pharmaceutical drug products and biopharmaceutical drug products, alongside you will learn, or you will hear, or you might have read about these two terms that are generics and biosimilars. Okay. Before getting into the details of generics and biosimilars, we need to understand that there are two types of medicinal products anytime available in the market. One is a patented drug, other one is a off-patented drug. Patented drug means there is an organization who has developed this particular molecule and manufactured, and they hold the patent for this molecule. Then it is a patented drug. When the patent gets over, then the other organizations can develop a very similar molecule, which is physiologically and structurally similar to the parent molecule, that is the patented molecule. This patented molecule is called as a reference listed drug, which means if you are producing a molecule which is very close, which is similar to reference listed drug, then you can call it as a similar therapeutic. If the similar therapeutic is a small molecule, you call it as generics. If the similar therapeutic is a large molecule, you call it as biosimilar. So basically, there is a reference listed drug, which is a patented molecule. And after the patent is over, patent period is over, the other organizations do research and come up with a similar drug, which is physiologically and structurally similar to the patented drug. If the new drug that is being developed, if it is a small molecule, it is called as a generic drug. If it is a large molecule, it is called as a biosimilar. That's the understanding we all need to have. Because when you start searching jobs in the portals, in the company portals, or into the job search sites, commonly you will come across with this term biosimilar and generics. So I thought it will be a good idea for all of us to first align in these two terms, generics and biosimilars. Okay. If this part is clear, then we will move into the next part, that is the different types of markets models, operational models in the market. When you say a drug, the one slide before, when we say the drug is basically composed of two major classification of molecules, classification of compounds. One classification of compound called as API, that is active pharmaceutical ingredient. The other portion is the excipient, that is the supporting ones, the molecules which support the functioning, delivery and functioning of the API. Okay, so these are the two compounds. These are the two cluster of compounds. Here, there are two types of industries operational. Has anybody said anything? Okay. So basically, there are two operational models in the industry. The first one is the API industry. The API industry is also called as the bulk drug industry, or it is called as drug substance manufacturers. This is the API one, which means they produce only API. They do not go one step ahead and formulate it. They produce API and they sell the API. The other portion is the formulators, who take the API from the API manufacturer, manufacturer and mix it with excipient and convert it to the final format in which the patient is consuming or the consumer is consuming. For example, if it is a paracetamol, the API manufacturers produce paracetamol powder. The formulation industry takes the paracetamol powder and mix it with the bulking agent, stabilizing agent, preservative, and so on and so forth, and convert it into a paracetamol tablet. The tablet is called as a format, final format, or they convert it to a injectable. Injectable is a final format. So these people are called as the drug product manufacturers, DP, or drug product manufacturers, or formulation industries. So these are the two major models in which the industries operate. Interestingly, there is a combination of these two are also available. There are some organizations who produce API, sell API, and they also produce the drug products from some of the API they have. For example, if an organization is producing paracetamol, this organization X, which is producing paracetamol, will sell the paracetamol to company Y, Z, and so on. At the same time, the organization X will also produce paracetamol tablets and paracetamol injections and sell it, which is absolutely allowable, right? So this is an interesting combination that is available in the industry. So in general, we can classify it as API, bulk drugs, or drug substance manufacturers, and formulation or drug product manufacturers, and a third combination, which is a API and formulation manufacturers. These are the market segments. Now we'll drill down to one step closer to these drug products. These drug products, either they are sterile, these drugs are either sterile or non-sterile in both stages. In API stage also, in the formulation stage also, it could be either sterile or it could be non-sterile. If there are certain cases, the API is manufactured as a sterile compound and sold as a sterile compound as a frozen drug substance, like what you're seeing in the picture, in small bags, in bioprocess bags, it will be sold as a frozen drug substance. Non-sterile API powders are sold in bulk, in drums and reams and so on, 5 kgs, 10 kgs, 50 kgs, 100 kgs, and so on. So it is sold in a bulk. These formulations are made from API, can also be sterile or it could be non-sterile. When I say sterile, some examples like, 1 minute, some examples like injectables. As you can see in the picture, most of the, is my slide visible to you?
Yes, sir.
Yes, sir.
Okay, wonderful. So formulation examples are injectables. Injectables are sterile formulations, and non-sterile formulations are nothing but tablets, capsules, syrups, which are coming under oral suspensions, or it could be like ointments and so on. Not only injectables are sterile, interestingly, the eye drops, ear drops, these are nasal sprays, these are all sterile compounds only, sterile formulations only. So these sterile formulations, as you can see in the picture, right? As you can see in the picture, the sterile formulations are made out of non-sterile API. I don't know if you are able to see this red color clearly, but I'll show through a pointer. Okay, so if you can see the cursor moving, the sterile formulations are made from non-sterile API. Very rarely, sterile API is used for manufacturing of sterile formulation, and always sterile API is used for manufacturing of non-sterile API is used for manufacturing of non-sterile formulation. This is the basic construct in the operational model in an industry. Okay. So this is how we need to understand the organization part of it. If you remember, we said ROI, IOR. We spoke about industry, which is pharma and biopharma industry, and sometimes the service industry. In this service industry, what type of organization you would like to work? Would you like to work in a manufacturing organization or a service organization? If it is a manufacturing organization, would you like to work in a bulk drug manufacturing organization or a formulators, drug product manufacturing organization? Within drug substance manufacturers, would you like to work in sterile or non-sterile? Would like to work in sterile drug product manufacturing, non-sterile drug product manufacturing, which means those who are manufacturing tablet capsules or those who are manufacturing injectables. This is how you should, I think you should, this is also one of the way to approach how do we get into a career successfully. If this model is clear, I would like to move to the next portion. Okay. This portion is very, very important. It is a little more technically intense. I would like you to patiently listen to me, and if you have any questions, please park it. We'll have a question and answer session after the talk is over. At that time, we can clarify. Okay. Functional domains. In every, this is exactly where you need to understand how to align your interest and your job opportunity and your personality trait. Every industry has a domain. For example, if it is a biotech drug manufacturer company, there are 4 domains available in these companies. Okay, these domains are research and development, manufacturing, quality, and regulatory affairs. These are the 4 broad domains. Please remember, I am talking only about the technical domains. There are other non-technical domains like HR or supply chain management, corporate communication, commercial, finance, all that are also available. Okay, so I am restricting my conversation completely to the technical domains. So in these technical domains, in each of these domains, there are some of the departments we should know. In research and development, as you can see here, there are 2 terms. One is a research, other one is a development. One is a research and other one is clinical research. Clinical research is called as the drug development pathway. Research is the discovery pathway. Okay, which means what are all the activities you are doing to discover a product that comes under basic research or the research, and after you discover, what are all the steps you are taking to make sure it is safe and effective for human consumption. That part is the clinical research part. That is the developmental part. That's why you always call it as R&D, research and development. In the same slide, I will tell you, this is my last slide also. In the same slide, I will tell you about in detail about how to understand each of these departments in detail. That portion also we will go through. In manufacturing, there are 2 major segments. One is MSAT and other one is production. Earlier, we used to have a department called as PD, process development, as a part of R&D and manufacturing that connects the R&D and manufacturing. But nowadays, manufacturing has a separate department called as MSAT, that is Manufacturing Science and Technology. This manufacturing science and technology plays a very, very crucial role in executing what R&D has developed in the large-scale manufacturing. We'll talk about it in detail. Then you have production department. Production department is the one which converts the raw material to finished products, right? So you have a raw material and they use equipments and processes, and human beings are involved in executing these procedures to convert the raw material to a finished product. Then comes quality. In quality, you have 3 subdomains: quality assurance, quality control microbiology. As you can imagine, if you are buying a medicinal product from the pharmacy, there are 2 things you are very particular. Number one, the product should be effective. When you're buying a tablet or any medicinal product, the first thing you are looking at is effectiveness, efficiency. The product should be effective for what you are buying to. Number one. Number two, it should be safe. So the quality domain makes sure that the product which is coming out of a pharmaceutical industry meets the potency and efficacy part when it comes to the consumer side. We call it as patient experience. The patient experience is ensured by the quality domain. In quality domain also, as I told you, there are three sub segments, which are the departments: QA, QC, and QCM. There is a difference between QC and QCM, which I'll elaborate. And the last domain is the regulatory domain. The regulatory domain is a very, very important domain. Without regulatory domain, I don't know how many of you are well aware of this existence of a regulatory domain in the pharmaceutical industry. Because most of the time the manufacturing and R&D are visible. To some extent, quality domain is very visible. This regulatory domain is not very visible, but at the same time, without the presence of this regulatory domain, the product cannot reach the market anywhere, whether it is in India or it is in the US or it is in the EU or it is in Japan or any other country you can think of. You can produce the product in your company only with the help of manufacturing license obtained through regulatory process. So number one, you need the support of regulatory agency to get the manufacturing license. Even if you manufacture your product in your company, without the help of regulatory affairs, you cannot sell your product within India or any of the countries. You cannot export to any of the countries without the regulatory process and approvals. So it is playing a very, very important and crucial role. So these are all the domains and these are all the departments. Give me 1 minute. I would like this sip water. Thank you. So we started off with the understanding of what is the industry, what are the different categories of industry, working models or operational models in the industry. And within one industry, within one organization, what are all the different domains available, and within each of these domain, what are the departments available. Up to here we have come. Now what we are going to do is we are going to details of each of the department. First, I would like to talk about the research department. When it comes to research department, the first and foremost thing that is focused on in a research department is the discovery. Whether it is a biosimilar or it is a novel molecule. Novel molecule means new compound, new drug. In the industry, we are using two terminologies. It will be a good idea if you know about these two terminologies. One terminology is called as NCE, other one is called as NBE. NCE means new chemical entity, NBE is new biological entity. So based on whether it's a pharmaceutical company or a biologic company, it depends what they are discovering. Are they discovering a NCE or they are discovering a NBE? The discovery starts with identification of a target. If you are a biological bioscience student, by now you would have understood what I'm talking about as a target. Target is nothing but a marker, biomarker, or determinant, antigenic determinant, a determinant, cell surface determinant. For example, CD4 receptor, these receptors are called as targets. Once the target is identified in a cell, first we'll take an example so that our discussion becomes very easy. For example, let's say a company wants to develop a drug against COVID. When I say COVID, SARS-CoV-2 virus. SARS-CoV-2 virus is going to affect the human being by attaching to one particular receptor in the lung cell, which is called as ACE2 receptor. That is the target, which means when you want to develop a drug molecule, you are not directly starting with the drug molecule, you are first starting with the target. ACE2 receptor, which is available on the lung cells of human being, is the target. Why we call it as a target is because the virus, SARS-CoV-2 virus, is going to attach that particular ACE2 receptor, which means if I can modify this receptor, if I can destroy this receptor, or if I can do engineering in such a way that if the receptor cannot be bound by the antigen from the uh, from the virus side. So we cannot have a control over the virus. What is in our control? In our control is the target. That is why we start with the target. So once the target is identified, we have to find out what are all the chemical and biological compounds which can attach to this target. This portion is called as high throughput screening. When you do a high throughput screening, you will get to know about the binding efficiency of various compounds to the target. Once once the number of molecules which can attach to the target is identified, then it is optimized. Optimized means proteomic work is done, little bit of proteomic work is done to increase the efficiency of binding. So step number one is binding. By binding, you can complete one activity called as target validation. Target is identified, target is validated, which means you have identified the target, structurally characterized the target through 2D structure, 3D structure elucidation is completed. You know about the target. Now the next step is to find out what are all the molecules, hit molecules or the medicines which can go and bind to the target. You are finding out the binding efficiency. Based on binding efficiency, you are calling certain hit molecules as the list of molecules. Once the hit molecules are optimized, they can be called as the leads. That is why you call the activity as a lead optimization. You are optimizing the lead in such a way to increase the binding efficiency of the hit molecule to the target molecule. Once this portion is done, the next step is upstream and downstream development. Now you need to identify in what organism you are going to express this. You can develop the clone. You can develop the clone in any of the intermediate expression system. You can develop the clone in E. coli, or you can develop the clone in whichever organism you want. But end of the day, you need to also understand, are you going to use, or am I going to use the same organism as the expression system, or I have some advantages of using some other organism as a expression system. For example, let's say monoclonal antibody. If you are having a clone which can produce monoclonal antibody, which means you have inserted the genes that are required to produce the monoclonal antibody in a particular microorganism. But when it comes to expression system, it is a good idea to express this monoclonal antibody in CHO cell, which is Chinese hamster ovarian mammalian cell. Because if you express it in mammalian cell, then the post-translational modification will be better. Now, this expression is called, or the expression system is mass produced, which is called as the upstream. So upstream is nothing but the product formation stage. The product formation stage is called as upstream. In R&D level, it is called as upstream development, which means in a small scale, you have to develop the process. When I say process, you have to say in what medium you are going to culture this expression system. How do you induce the expression system? How to monitor the parameters like pH, dissolved oxygen, temperature required, all are optimum conditions. How are you going to monitor? This entire development is called as upstream development. Once the product is formed, then the next step is to remove the product, recover the product from the biological system, from the cell, and purify and concentrate this product and produce it as API. The purified concentrated product is nothing but the API, the active pharmaceutical ingredient, which we spoke about in the earlier slide. Okay, that portion is called as the downstream development, which means recovery, purification, concentration method comes under the downstream. So again, the small scale work that is done to develop the parameters are called as downstream development. So upstream development, downstream development. If you can imagine, any of the students who are listening to this talk, if you are from biology background, By now you would have understood that you have a very strong job opportunity in R&D if you know about the biological system. Imagine, can a chemical engineer or a mechanical engineer can ever, or even a computer science engineer, can never imagine how to express a protein in a cell because they don't even know the cell cycle. They don't understand the cell cycle. So essentially, you can do a IT job, but not the other way around. So my sincere invitation to all of you is that please keep your interest and passion intact and continue to look for a job till the time you land up in your dream job. Only thing I would like you to focus here is the departments are posing a very strong technical job roles. Job opportunities are available if you know and get trained in the areas whatever we are discussing here. So that's the upstream and downstream development. When you are doing the upstream development and downstream development, one of the important thing we need to concentrate is the critical parameters. What are all the critical activities which you are doing, which will allow you to get the goalpost? Goalpost means in how many trials you are able to get to the optimum condition. What is the optimum condition? How are you getting to that? This how part is very important. That's the goalpost area because the how post part is the one which is going to define the process. It is not just an inoculation of mammalian cell culture in a DMEM medium and allow it to grow. It is not going to work that way. How you are going to agitate it, in what RPM you are going to agitate it, at what pH you are going to maintain it. Because when you are maintaining certain pH, over a period of time, some of the secondary metabolites will form and pH will drop down. When the pH is dropping down, how are you going to increase the pH? If the temperature drops down, how are you going to increase the temperature? If the dissolved oxygen drops down, how are you going to increase the dissolved oxygen? That's the goalpost, how part of it. And then you are going to define what are all the optimum parameters in which it will work. These are called as critical quality parameters, which determines or defines the key product profile. If the product has to have this much of purity, this much constant percentage of purity, it is very important if you focus on key quality parameters, critical quality parameters like pH or temperature or dissolved oxygen or impurity. How are you going to handle the secondary metabolites, impurities, and so on, right? Then how to qualify these methods? You are trying to develop certain methods to monitor the parameters. For example, pH determination, temperature determination, or assay. For example, when you are taking a sample from the bioreactor and finding out the titer, what is the protein concentration, then you are using different methods. You are using electrochemical methods, you are using biochemical methods, you are using electrophoretic methods, spectral methods, you are using different methods. So all these methods has to be qualified to make sure that the method which you are using is suitable for the activity which you are doing. That is called as method qualification. So that's a part of the research. The most important part of the research is to find out how long the product can sustain. This is the one which is defining the shelf life of the product. The stability test is conducted to determine the shelf life of a product. The stability test is done in two ways: accelerated stability study, ongoing stability study. A little bit of accelerated stability study is done in the R&D level, and later when it comes to manufacturing, the quality control will continue to perform the ongoing stability test to make sure that the product uh, can claim certain amount of shelf life or expiration date. Based on all this, the R&D will give the specification and limits. Technical specification for the product, that is the product profile, which should also align to the pharmacopoeial requirement. In pharmacopoeia, if you are familiar with this term pharmacopoeia, United States pharmacopoeia, European pharmacopoeia, in pharmacopoeia there is a separate portion called as monograph. This monograph gives some idea, a brief idea about the product profile. So the product profile that is created by the R&D should be in alignment with the product profile that is outlined by the pharmacopoeia monographs. And limits also, the internal limits are set by R&D, external limits, the quality limits are set by the pharmacopoeia. Once all these things are done, then this technology is transferred to manufacturing. Now the research portion is over, the manufacturing has to take over. If you are wondering when the manufacturing will start, will the manufacturing will start only in a commercial scale? The answer is no. Interestingly, the manufacturing starts when the clinical research is going on itself. Because for you to conduct clinical research, you need the product, which means you have to manufacture the product. So at the time of clinical development itself, we have to start manufacturing the product and start giving the product to the R&D department. R&D will conduct two types of clinical research. One is a preclinical studies, other one is the clinical trial. Preclinical studies are done on the animal models. In animal models, the preclinical trials are done in order to find out the toxicity profile. When I say toxicity profile, safety, to establish safety. It is called a safety study. Technically, it is called a toxicology study. Toxicology study is done to determine what are all the side effects that the product can create in the animal model. And is this side effect acceptable within the limits from the pharmacopoeia perspective? Because end of the day, when you are submitting this information to the regulatory agency, they should be in a position to approve it. So upfront, you have to find out what are all the toxicity, this or the side effects this product can produce in the animal models. When I say animal models, close to the target animals. If you are talking about human, generally it is done on mouse because mouse is the closest mammalian model to the target that is human being. That's the preclinical studies, that is the animal study focused on toxicology. The second portion is the phase trials. Phase trials are done on human volunteers. In phase trials, there are phase 1, phase 2, phase 3, and phase 4. Phase 1 clinical trial, phase 2 clinical trial, 3 and 4 clinical trials. In phase 1 clinical trial, the uh, once the regulatory approval you are getting for your IND, which is investigational new drug, IND, then you can move forward to start your trial on human being. When you're starting the trial on human being, the first trial is done on healthy volunteers. If I take back the same example what I took in the research site for the target, if I'm trying to understand or if I have an hypothesis saying hydroxychloroquine can be used for the treatment of COVID disease management in human being, if this is the problem statement I'm creating, what we need to do is in phase 1 clinical trial, the healthy volunteers who are not suffering from COVID disease has to be recruited, and this medicine, hydroxychloroquine, for example, is given to these people and find out if there are any side effects coming up. And I say side effect coming up, it will come up. But what we are trying to understand is the same side effect what is observed in the animal trial is happening again in the human trial, in the phase 1 clinical trial also. That's all is the comparison done. In phase 2 clinical trial is the place where actually you are doing the proof of concept. Your concept is, can hydroxychloroquine be used in human being for treatment of COVID? Which means in phase 2 clinical trial, the deceased patients are recruited, which means COVID affected patients are recruited and the medicine is given to them to find out any clinical improvement is there, clinical correlations can be made so that there is a logical endpoint to the medication. So that is the phase 2 clinical trial. In phase 2 clinical trial, one more important activity done is the dosage adjustment. Whether it has to be given as 200 mg molecule or can it be given as 2 into 100 mg in 2 hours, 3 hours. This is the dosage adjustment studies. So basically in phase 2 clinical trial, two things are very important objectives. Number one, efficacy. How effective the product is on the disease in alleviating the disease. That is the first objective. Second is in what dosage it has to be given based on the stage of the disease. That is also studied as a part of the phase 2 clinical trial. Phase 3 clinical trial is a little more extension of a phase 2 clinical trial. Nothing new is being done, but only different types of human population is taken into consideration. We call it as a ethnogenicity. Different ethnogenicity of human being is considered into the clinical trial. So it is little more elaborate clinical trial done on human beings, which is phase 3 clinical trial. Phase 4 clinical trial happens after the product is manufactured commercially and made available commercially, and people have started using this product in the market after 1 year or after a year and a half. Then the post-marketing surveillance is taken, which is called as the phase 4 trial. The objective of the phase 4 clinical trial, which is post-marketing surveillance, is called as pharmacovigilance. Pharmacovigilance means identification of adverse drug effect of the product. Because in the clinical trial, you might have done in certain thousands of patients you might have done. But when it is commercially sold, we really do not know how many thousands of people are going to consume this product, or lakhs of people are going to consume this product. In these lakhs of people, is there any adverse effect? Adverse effect means any serious side effects has happened in the population. That study is called as pharmacovigilance. Please remember, people with life science background, MSc or MTech or BPharm or MPharm, MTech or any of these bioengineering, these are the candidates who are very, very useful in conducting pharmacovigilance activity and the analysis of data which is coming back from the pharmacovigilance on the ground also, and the background work also, that is desk work also. It is preferred. The bioscience students are preferred in this. For this, you have to monitor the job opportunities coming from CROs, contract research organizations. In the last talk, I told you examples of CROs, like it could be, I don't want to promote any names here. You can search for contract research organizations in India who are conducting clinical trial and clinical trial data management. You will get a series of organizations who are into it. Okay, so that's the R&D part and the clinical research part, the R&D part. The second portion is the manufacturing. In manufacturing, I told you there is a separate department called as MSAT, which is Manufacturing Science and Technology. In Manufacturing Science and Technology, what they do is they do the scale-up studies. Scale-up studies means R&D has completed their studies in the lab scale, 1 liter to 5 liter scale. Now, if you want to suddenly convert this process and execute the process to 2000 liters or 20,000 liters, you cannot use the same parameters. You may have to conduct certain studies in a pilot scale and then take the data and put it onto the process scale. This portion is called as the scale-up studies, which is done from the tech transfer document, which is provided by the research department. As you can see here, tech transfer is the last portion of the research department, and that is the input for the manufacturing. Okay, so once they complete the scale-up studies, once the process is validated, then they do the process validation to validate the process. Once the process is validated, then it is given to the manufacturing. The production department conduct the upstream. When I say upstream processing, either it is a fermentation or it is a cell culture. In biological world, either you use mammalian cell lines or eukaryotic cell lines, or it is a microbial eukaryotic cell line or a prokaryotic cell line. For example, it could be bacteria, or it could be yeast, or it could be CHO cell, Vero cell, BHK21, and so on. So these are all the different cell lines that are used. So based on the cell line, either you are doing a fermentation or they are doing a cell culture. So that is the upstream part of it. As I already told you, upstream is nothing but the product formation stage. Once the product is formed in the large scale, you have used a 20,000 liter bioreactor. In that, you have completed 18,000 liter working volume, and you have completed your fermentation or a cell culture. Product is formed. Induction is done. Enough time is given. Product is formed. Now the next step is downstream processing. Downstream processing starts with the recovery of the product from the bioreactor or the fermenter. Okay. Once the product is recovered, technically it is called as a harvest. Once the product is harvested, then for harvest also you are using different methods. It could be continuous centrifugation, or it could be disk filtration and so on, stack disk fermentation and so on. Once the harvest is done, then the next step, as I told you, it is purification and concentration methods. It could be primary purification starting for capture chromatography. After capture chromatography, it will go through ion exchange chromatography. After ion exchange chromatography, it will go through hydrophobic interaction chromatography. In between, there will be a buffer exchange, and finally, there will be a concentration. Buffer exchange and concentration is through a technology called as TFF technology, tangential flow filtration technology. So based on that, you can do the buffer exchange or the concentration. Once that is done, you have a concentrated API in place. Okay. Once the API is ready with you, then you do the formulation. As I told you, you have two options. The organization has two options. One, sell it as API itself. One option is convert this API to a powder, a crystallized powder, lyophilized powder, whatever way you want. You convert it to a powder and sell this powder. Option A. Option B, you can do the formulation. You can mix this powder with excipients in water. Water as the solvent. You can mix it or any other organic compound as a solvent based on the solubility factor. You can solubilize this and add excipients to it and convert it to a formulation. If it is a sterile product, then there is a sterile filtration. As I told you, my entire discussion is focused on biopharmaceuticals. That is why I'm focusing on sterile filtration. Sterile filtration is the sterilization method for any biological drug compound which has heat sensitive compounds like proteins, amino acids, and so on. So essentially, all the biological drug products has to go through sterile filtration. So when it goes through the sterile filtration, you will get a sterile filtrate. Once you get the sterile filtrate, this sterile filtrate is filled and finished into vials, ampules, whatever way it is. So that is the fill finish. In fill finish, there are different types of machines: vial filling machine, blow fill seal machine, form fill seal machine, and so on. So these are different types of fill finish technologies that are available through which you can complete the production activity. Once the production activity is done, in between the production activity also sample is taken. Okay. First, let us see QA. QA has three major roles. Number one, whether it is a manufacturing process or testing process, QA has to approve and validate the method. Whether it is a manufacturing method or a testing method, everything has to be approved by quality assurance. The second major activity of quality assurance is QMS, quality management system. If you have already completed your graduate or postgraduate degree in biosciences, and if you are wondering what are the areas I can upskill myself to get into quality domain, this is one of the area. You can do certification programs in quality management system and so on. So quality management system is nothing but implementation of GXP. For example, implementation of GMP processes, implementation of GLP process, implementation of GCP processes. GMP refers to good manufacturing practice, GLP refers to good laboratory practice, and GCP refers to good clinical practice. So implementation of all these together is called as the quality management system in the quality domain. The third important one, as I told you, quality is the one which takes sample from the production at the time of production as well as after the completion of fill finish. All these samples are taken to the quality control department. The assay is done, impurity profiling is done, and stability test is done. Based on the assay means how much pure the product is. Impurity profile is how safe the product is. If the impurities are within the acceptable limits as per the pharmacopoeia limits. Based on that, they will give input to quality assurance. As you can see here, in the third point of quality assurance, lot release. Based on all the data provided by manufacturing, which is called as BMR, batch manufacturing record, and which also includes the quality test reports. Based on that, quality assurance take a decision whether to release this batch or not. That's called as lot release. So quality control's major role, quality control analytical, kind attention to MSc chemistry, MSc biochemistry students. So the quality control analytical lab is involved in sampling, assay, impurity profiling, and stability testing. As I already told about stability testing, I'm not repeating here. Quality control microbiology, very, very important role they play. If you are studying microbiology as a subject, BSc microbiology, MSc microbiology, or whatever, or a subject in as a semester. For example, if you are a MSc biotechnology or B Pharm or M Pharm, whatever it is, and B Tech or whatever it is, and you are studying one semester microbiology, still you hold a chance to get into quality control microbiology department, where environmental monitoring is done. Environmental monitoring means the environment in which the product is manufactured and the product is tested in the quality control department. This environment is monitored for its microbiological attributes by the quality control microbiology department. Environmental monitoring includes the air monitoring, surface monitoring, and personal monitoring. Monitoring of the human being who are involved in the manufacturing or the testing. The second is the bioburden testing. For example, you are using a lot of non-sterile intermediates for the sterile manufacturing. Non-sterile intermediate means there is IPQC sample, in product quality control sample. When the product is manufactured, you are taking some of the samples. Those samples are tested for bioburden. And sterility test. As I told you, most of the biologic products are injectables, so they are sterile in nature. So the quality control microbiology conduct a test called a sterility test to find out whether the product is sterile before the product reaches the consumer or the patient. And finally, they are also doing the bacterial endotoxin test to find out whether the biological impurity is within the limits or not. Endotoxin is nothing but the lipopolysaccharide layer of a gram-negative bacteria. So it has to be in the lowest limit as per the regulatory guideline. That's the role of quality control microbiology. And finally, regulatory affairs. In regulatory affairs, there are very, very important activities done, which is creation of the drug master file. The drug master file is the one that is submitted by the drug manufacturers to the regulatory agencies. United States Pharmacopoeia is, the US FDA, United States Food and Drug Administration, or EMA, European Medicinal Agency, or PDA, PMA for different countries. You have different regulatory agencies. These regulatory agencies has to receive the DMF from the drug manufacturers in order to give them a marketing authorization based on the data that they are providing. In addition to that, you have to send the DMF to the state regulatory or central regulatory agency of your own country to get the manufacturing license, where your manufacturing plant is set up, is located. So basically, the filing is done for two different purposes. One to obtain the manufacturing license and another one to obtain the marketing authorization. Please remember, I am giving a very bird's eye view of what are all the domains. There are so much more into it. Please don't imagine this is all the biopharmaceutical departments are doing. Okay, so these are all the departments, technical departments that are available in a biopharmaceutical industry. So as I told you in the first slide also, first slide itself, I would like you to focus your career in this order. What is the industry in which I am working? Am I ready for that? Am I equipped for that? What is the organization I wish to work? Do I know about the organization? What are the work that the organization is doing? Where and all it is located? About the I know the information about the organization? Which role I would like to work? Do I want to work as a research person or in a CRO? Or do I want to work as a production person, quality person, or a regulatory person? What role I would like to work? In this order, if you can align your career search, I'm sure all of us will come out with flying colors. Thank you very much. I would like to pause here and open up the session for questions. So how do I go about? I'll do one thing. I'll stop presenting so that. Okay. All right. Yes, there was a case of self. Yes, of course. So as Jivita Nasir was mentioning, there are so many cases already available. So Ranisha, thank you for the informative session. May I ask what does process R&D refer to, upstream or downstream? What does process R&D? Process R&D is nothing but both upstream and downstream, upstream development and downstream development. That's called as process R&D because both are processes. It is called as DSP and USP, upstream process and downstream process. Is a bioprocess engineer can join R&D? Of course, yes. There are a lot of MTech candidates who join into R&D for specific developmental work. You know the different opportunities in regulatory departments. That's exactly what I was talking to you in the last portion of the presentation. DMF or filing or regulatory submission preparation, regulatory reviews, and so on. So what's the difference between sterile and aseptic? I know who is asking this. Kishore. Kishore, sterile is the complete absence of life in a product. Aseptic is a condition in which absence of microorganism in a defined area. If you say less than 1 CFU per meter cube, it is aseptic. If you say absence of microorganism in 1 meter cube, it is called a sterile. Yes, biochemistry can join R&D. Most informative, good session. Thank you very much, Ramachandramurthy. Jivita, you explained everything so well. Thank you for the session. You are welcome. Sir, could you say a few ways to move into, homemaker into clinical trial and pharmacovigilance? Yes. So you have to start focusing on understanding the post-marketing surveillance, clinical data management. You may have to look into some of the statistical tools, like how best you can use the Excel, SPSS software, SAS, SAS certification. That will gradually take you closer to your clinical trial and pharmacovigilance. You can do a diploma in pharmacovigilance and so on. What is the duration of pharmacovigilance study? It depends. It depends on the type and nature of this, because sometimes the pharmacovigilance is initiated by the government. If there is a strong signal, I have not given you the full story of the pharmacovigilance. That is why this question is originating. So it depends. The timeline completely depends. Is the drug sent back to R&D or reform? No, there is nothing called as reformulation. It's a research basically to find out what has gone wrong. It is again completely a fresh research that is taken. Can freshers directly enter into QC microbiology? Yes, you can. MSc Biotech student can apply for MSET. Absolutely, yes. Or how can I go into regulatory department biotechnology after doing masters in biotechnology? The regulatory affairs department is a little, you have to approach it little carefully. Only with the postgraduate degree, it is very difficult to join. You may need to upskill yourself through some of the certification programs, like in Biocon Academy or in RAPS program and so on. Then it will be easy for you to get into regulatory. Oh, sorry, I'm missing so much into this. Give me a second. Is biotech enough? Should we need to pursue? How to join Biocon Academy? Yes, you can go to bioconacademy.com, find out what are the courses for the students, find out which course you are aligning to, and you can join. Be biotechnology engineering student in current industry. Of course, there are a lot of opportunities in production, in different developmental work, even in QA validation. There are a lot of roles available for B students also. Can someone who does MBA, where is it gone? After MSc Biotech, get a job in company? Yes, absolutely. When it comes to marketing, market intelligence, business development, A strong MBA and MSc Biotechnology is a very good combination to have. Thank you for very informative. Can I MSc license integrated? Yes, there is no difference between a individual or integrated course. MSc is MSc from industry's point of view. From industry's point of view, degree, more than degree, what is the skill you are bringing in? That is more important. MSET. Is there a course available for MSAT in bioeconomy? That's an interesting question. Snehal, there is no separate course, but the bioscience course addresses the MSAT also. We have a bioscience course which addresses MSAT requirement also. So if any, I can approach you in future and how may. Please send an email to admissions@biocon.com addressing to me. I'll respond to you. So being a fresher who complete the BTech in biotechnology department. Yes, in entry level, certainly yes. And when it comes to improve the experience in further, it is not only the experience, but also skilling. After joining, what are the things you are doing for yourself to upskill yourself? That determines your improvement within the organization. Then Tamil Rasan, sir, clinical data management work is compulsory to do. No. It is not compulsory. The problem here is if you are a biotech student, your resume is going. If there is another biotech student, his resume is going or her resume is going, along with the certification in SPSS or SAS, then they stand a better chance. So doing a certification is not compulsory. It is good to have to be competitive in nature. Oh, there's an internal discussion also. Akshaya saying thanks to Jivita. Very good, excellent. Joseph, sir, could you please give your. I am not a person to give you opinion on KG certificate program. You can search for alumni in LinkedIn who have completed the KG certificate program, connect with them and check with them. Our alumni can speak about the program much better than us. Thank you very much for the insightful session. Thank you very much. Okay. Very good. So we are crossing the time of 6:30. So what I will do is I'll pause here and we will all meet in the next technical session. If any spillover questions are there, we'll try to address it. Thank you very much and have a very good evening. Bye to all of you.
Thank you, sir.
Hi, sir.
No problem. Bye.
Thank
Thank
you,
you,
sir.
sir.
No problem.
Thank you, sir.